Hi everyone, so today we're going to be looking at meiosis, and so the study design dot point is the production of haploid gametes from diploid cells by meiosis, including the significance of crossing over of chromatids and independent assortment for genetic diversity. So let's get started. So meiosis is a little bit different to mitosis, which we've been over, in that it produces genetically unique cells. So each of the 4 cells that are produced at the end of meiosis are genetically unique to each other. And they are haploid cells, so they have half the amount of genetic material as the cell that they originated from. And this only takes place at certain times in an organism's life cycle, so it doesn't happen all the time the way that mitosis does, and is specifically involved in sexual reproduction. So let's look at a video that describes or shows the process of meiosis. So as I said before, meiosis is a type of cell division that's required for sexual reproduction. So it produces gametes, and that's sperm and eggs in humans. It only contains 1 copy of each chromosome. This is important because you only pass on half your genetic material to your offspring or your children, and so your gamete will combine with someone else's gamete, so an egg and sperm will combine together, each containing only 1 copy of each chromosome, and when they fuse together to form a fertilized egg, then we have 2 sets, so 2 sets of chromosomes that is diploid and is ready for the development of an embryo. So meiosis has 2 distinct cell divisions. So before meiosis actually occurs, we begin with a diploid germline cell. So germline just means the cells, or the cell that the gametes come from, so that the original cell is called a germline cell. Diploid, as you should remember, means 2 sets of chromosomes, and humans have 2 sets of chromosomes. Then we have meiosis 1, which is the first cell division, and this separates homologous chromosomes, and so there's only 1 copy of each chromosome in each cell. But remember, they are still sister chromatids, so there's 2 chromatids attached together. And at the end of meiosis 1, these 2 haploid cells go on as inputs for meiosis 2. And so at the end of meiosis 2, we end up with 4 haploid gametes. So 1, 2, 3, 4. These sister chromatids separate, and there's only 1 copy. and one single chromatid in each cell. So here is a list of all the different stages we're going to go through in terms of cell division. As you can see, we have meiosis 1 here, which still has prophase, metaphase, anaphase, and telophase, same way that mitosis did, but we put a 1 after it to indicate that it's part of the first cell division. And of course, before that, we have interphase. Then between meiosis 1 and meiosis 2, we have cytokinesis, which can also be called interkinesis. And then meiosis 2, we have prophase 2, metaphase 2, anaphase 2, telophase 2, and finally we finish with cytokinesis. So we're going to go through all of this in detail, and you need to know what happens at each of these stages. So during interphase, we know that this occurs before meiosis. The cell is preparing to divide. We have organelle replication and cell growth, same as we do in mitosis. And the chromosomes replicate to form 2 genetically identical sister chromatids. So we're starting off with these single chromatids. They replicate to form 2 sister chromatids. So now we have the start of meiosis 1. So the first stage is prophase 1. So we have the nuclear membrane breaking down, the chromatin is condensing into chromosomes, centrioles migrate to opposite ends of the cells, the spindle fibers form, and the chromosomes form homologous pairs, which you can see right now, and they undergo crossing over. So crossing over is a naturally occurring process which produces chromosomes with new combinations of genetic material. So once the homologous chromosomes pair up, what happens is that parts of the homologous chromosomes cross over, like you can see over here in this diagram, and the point at which they cross over is called the chiasma. So the DNA strands from the chromatids are cut at this position. Then those equivalent, or the segments of the equivalent DNA is exchanged. So you can see part of the blue chromosome went onto the red and part of the red went onto the blue. The DNA reconnects at that exchanged piece, and now we have recombinant chromatids. So the chromatid that has exchanged a piece of DNA is called a recombinant chromatid. The other chromatid, which has not exchanged a piece of DNA, is called a non-recombinant chromatid, because recombination is that it has literally recombined the genetic material. So crossing over produces recombinant chromatids, and this whole process is called recombination. And this results in greater genetic diversity of genetic material at the end of meiosis. And so chromatids that the haploid daughter cells inherit will no longer be identical. That's why each cell that we end up with at the end of meiosis is genetically unique, and this is one of the reasons. So now we have the second stage of meiosis 1, which is metaphase 1. So the homologous chromosomes line up in pairs randomly along the equator of the cell, one member of each pair on either side, as you can see is happening now. The process of them randomly aligning is called independent assortment, which we'll go into in just a second. Then the spindle fibers attach to the centromere of each member, so each chromosome. So as I said, when the homologous chromosomes line up randomly along the equator, this is called independent assortment. And so which side of the equator each member of the homologous pair ends up on is completely random. So we might end up with all the maternal chromosomes on one side and all the paternal chromosomes on the other. Or we could have one maternal chromosome and then some of the paternal chromosomes, or we could have a mix or a different mix. So the alignment on either side of the equator is completely random, and this again adds to genetic diversity. This is because the chromatid that is inherited in the daughter cells at the very end is completely random. And again, this is important for genetic diversity. And so now we have anaphase 1. So the spindle fibers draw each member of the homologous pairs to opposite ends or opposite poles of the cell. It's important to note that for each chromosome over here, they're still sister chromatids. And so only the pairs are separated. The sister chromatids are still attached to each other. And we have telophase 1, so the nuclear membrane begins to reform, the chromosomes decondense, and a cleavage furrow forms. That is the cell pinching inward. So now we are done with meiosis 1 and we have cytokinesis. So we know this is when the cytoplasm divides. And at the end of this, we have two separate haploid cells. So we do not have two sets of chromosomes anymore. We have only one set of chromosomes in each of these cells. However, each chromosome still has two sister chromatids. There is a period of rest before meiosis 2, so no DNA replication occurs. So we don't go through interphase again. We just go through cytokinesis, and then the next stage we will enter meiosis 2. So the cells at the end of meiosis 1 go into meiosis 2. So the first stage of meiosis 2 is prophase 2, so the nuclear membrane breaks down again, the chromatin condenses into chromosomes, centrioles migrate to opposite ends of the cell, the spindle fibers begin to form, and then we go into metaphase 2. The chromosomes line up along the equator of the cell, as you can see here. The spindle fibers attach to the center of the chromosome, so this is similar to what we have gone over before, and we enter anaphase 2. So the spindle fibers pull apart the sister chromatids and pull only one chromatid to opposite sides of the cell. And we finish meiosis 2 with telophase 2. So the nuclear membrane reforms, the chromosomes decondense, and the cleavage furrow forms. So that's the cell pinching inwards. And as you can see, we end up with 4 cells. So at the end, of course, we go through cytokinesis. This is not part of meiosis, this is where the cytoplasm divides. And we have 4 separate haploid cells. So each of these cells has only one copy of each chromosome as a chromatid, as you can see here. This produces 4 genetically different gametes. Right? So remember, we had crossing over, which exchanged segments of DNA, and we had independent assortment, and we only have half of each, so half of your chromosomes in each of these cells. So they are very different, they are unique cells. This is a summary of all the stages we've been through. I want you to know each stage of meiosis 1 and meiosis 2, what happens before, in the middle of these 2 stages, and after. You need to be able to describe all of this. So let's compare mitosis to meiosis. So we know that meiosis produces genetically unique cells, and these cells are haploid cells, so only one set of chromosomes in each of these cells, and they only take place at certain times in the organism's life cycle, and they're involved in sexual reproduction. Whereas mitosis produces genetically identical cells, and those cells are diploid, so they have 2 sets of chromosomes in each of these cells, and mitosis occurs throughout the organism's entire life and is involved in asexual reproduction, but we're not going to that now. Just want you to be able to compare mitosis to meiosis and know the differences between these two. So we've been looking at meiosis in animals. However, meiosis does also occur in plants, because plants can undergo sexual reproduction, specifically flowering plants can. And this involves both meiosis 1 and meiosis 2, as we just learnt. And so in plants, gametes are a little bit different. So the male gametes are the pollen grain and the female gametes are the ovule. So this contains the egg. Fertilization occurs in the ovule, and a diploid zygote, so a zygote is a fertilized egg, is formed, which is a seed. This is what we know as seeds. So seeds are the zygotes, or the product of sexual reproduction in plants. So for plants over here, you can see highlighted here is the pollen, so that's the male male gamete that is taken either by a bee or some sort of other insect or the wind to the female, to another plant, to the female part of the plant, where it goes down the pistil here into this ovule, which is highlighted, and there we have the growth of a seed. And that seed can then grow into a new plant or the offspring. So meiosis can go wrong sometimes, and when meiosis goes wrong, we call this non-disjunction, so it's an error, and this is when chromosomes do not separate during meiosis. In meiosis 1, if homologous chromosomes are not separated during anaphase 1, we can have an abnormal number of chromosomes at the very end of meiosis. So in meiosis 1, if those homologous pairs aren't separated, as you can see here, the two red chromosomes were pulled to the same side. At the very end, we have two cells with missing chromosomes and two cells with extra chromosomes. Or it can also happen during meiosis 2 if the sister chromatids are not pulled apart during anaphase 2. So here, if these sister chromatids are not pulled apart, again, we can have two cells with an incorrect number of chromosomes, and we can have two cells with a correct number of chromosomes. So this can really have a big effect on offspring in that we're producing gametes with missing or extra chromosomes. If the fertilization occurs with these incorrect gametes, then the zygote will have an incorrect number of chromosomes, which is called aneuploidy, which we've looked at. And so this is how aneuploidy occurs. So whether we have monosomy or trisomy, it is the non-disjunction in meiosis that causes this. So meiosis is super important for genetic variation because all 4 gametes produced by meiosis has its own unique combination of alleles. So these gametes are not the same. And offspring are always genetically different from their parents. And this increases genetic diversity in the population because each gamete is different because of crossing over and because of independent assortment. Offspring can look different as well. This is why you probably look a little bit different to your siblings. You might have some features that are similar because you both have the same parents, but for the most part, you look pretty different. And this is because of crossing over, recombination, independent assortment, and gametes being haploid. So the different combinations of the haploid egg with the haploid sperm, which I think is pretty cool, and it makes humans and other animals very genetically diverse. So as usual, I'm going to leave you with a meme. I liked both of these so much that I couldn't pick, so you can have both of them. Enjoy them, and as usual, please bring your questions to the class, and I will see you there.